Thank you for the great opportunity. This talk is to report data from our studies on the novel clinical relevant graft-versus-leukemia GVR model in humanized mice. This work was recently published in Journal of Leukocyte Biology. So first, just a couple of sentences regarding the disease we are dealing with, acute myeloid leukemia, AML. This is the most common acute leukemia in adult. In US, there's over 20,000 new cases was diagnosed each year. The median age at the diagnosis is 68-year-old. So pretty much this is elderly patient's disease. When patient had initial presentation, majority of them had the poor performance status and other comorbidities, preventing them to get intensive chemo treatment. So this is one of the challenges in the treatment of this disease. Overall, the prognosis is poor with 5-year survival of only 28%. AML treatment generally involve induction chemotherapy followed by consolidation. For patients get into complete remission, and for those with low risk in their leukemia features, we typically observe, and then hopefully a good portion of these patients' population can achieve long-term survival without further intervention. However, for patients with high-risk features of their leukemia, allogeneic stem cell transplant is likely the only curative way for the treatment. This is a high-risk procedure, but even after allotransplant, patients' leukemia can still relapse, and this is listed as the biggest challenge in the field. This is a summary of the overall process of allogeneic stem cell transplant. We first search a donor who has to be HLA match or at least a partial match with the recipient patient. These days, most centers use G-CSF mobilized cells, peripheral blood, as a source for the stem cells. So basically, we give donor G-CSF shot and to mobilize the stem cells from their bone marrow to peripheral blood. After a process called leukapheresis, we collect the cells, including enriched HSC, hematopoietic stem cells, and other cells in the blood. At the same time, recipient patient receive chemotherapy together plus minus radiation. This is a conditioning regimen to wipe out marrow to leave the space for new coming donor cells. After the transplantation, the recipient cells gradually go down and donor cells goes up. At about a couple of weeks after transplantation, we have a majority of patients achieve WBC engraftment. And about 1 month post-transplant, there's a 99% donor in chimerism study in the blood. Clinical benefit of allotransplant greatly rely on a process called graft-versus-leukemia, GVR effect, in which donor immune system, mainly the T cells, sees leukemia as a foreigner and kill the residual leukemia. In this way, preventing the leukemia relapse post-transplant. However, the donor T cells can also attack normal tissues in recipient patients, causing a problem called GVHD, graft-versus-host disease. How to maintain a good GVR without causing GVHD has been a long-standing challenge in the field. In the last decade, our ability to control the GVHD has some improvement thanks to several newly developed immunosuppressive agents and optimized immunosuppression regimens. However, not much has achieved in GVL field. So leukemia relapse post-transplant remain the top cause of the death in patient. This is the data from CIBMTR, listed the causes of the death post-transplant in the last several years. This is a summary. So we can see that 29% of the deaths within 100 days post-transplant, and 57% of the deaths beyond 100 days contribute to the deaths post-allogeneic stem cell transplant. So this is a very, clearly this is an area with a urgent unmet need. So with this, we become interested in building an optimal GVR model. So this slide shows the rationale of our study. So as mentioned before, clinical benefit of allotransplant greatly relies on GVR effect. AML relapse AML relapse remains the top of cause of the death post-transplant. The mechanism that mediates immune escape of leukemia during allotransplant, in other words, GVR failure, remains poorly understood. So studies in preclinical models are the key to define the mechanisms and discover new treatment. However, the lack of optimal clinical relevant GVR models is a major gap in the field. This precludes the move of novel treatment towards the clinical applications. Current experiment models to study GVR mostly build on the engrafting mouse leukemia cell lines into mouse model of allotransplant. So this is limited in clinical translation as many biological features are different between human and mice. For example, mouse leukemia cell lines are homogeneous and grow rapidly. They do not reflect human AML that are pretty much heterogeneous and grow over a long period of time. Second, in contrast to clinical allotransplant, where the donor and recipients are usually HLA matched, many mouse models of allotransplant use MHC mismatched pairs. Also, some leukemia antigens and clinically used drugs are human-specific, making it impossible to test these drugs in mouse model of leukemia. Humanized mice is a very useful tool for cancer research. So this built on immunodeficient mice engrafted with functional human cells, including human immune system. Two types of humanized model has been developed for immuno-oncology study. First is transient humanized model. So this model is built by injecting mature lymphocytes into the immunocompromised mice. The mature lymphocytes mostly derived from human PBMCs. So the limitation for this model is the xeno-GVHD. So this severe GVHD occurs in almost all the mice after a few weeks of the injection. So this made the therapeutic and study window very narrowed. The next one is stable humanized model using the CD34 positive human HSC hematopoietic stem cells to build. Stable humanized mice has been developed in last almost 20 years. Here are the major reports in the field. Immunocompromised mice, including NOG, BRG, NSG, and most recently Mr. G mice has been used. For HSC source, cord blood is the major source here. In addition, people starting to use G-CSF mobilized PBMCs. So these stabilized humanized mice built by all these groups all showed adequate human immune systems build up, so provide a very solid base to study human immune response. So with this, we hypothesize that if humanized mice is built by engrafting the HSCs derived from clinical donors of the allotransplant, then further injection of the patient-derived AML cells in these mice may enable the interaction between the donor immune system and patient-derived AML, therefore establish a clinically relevant GVR model. This is a schema for our study design. We use NSG mice. Human cells used here, are transplant donor-derived GCSF mobilized PBMCs. We purify HSCs using magnetic beads from these cells. Another human-derived cell is AML cell derived from the AML patients. An important point here is that we select HLA partial matched pairs between HSC and AML to prevent the rejection. So basically, three weeks old NSG mice got irradiated, sublethal irradiated before injecting with the HSCs, human HSCs, waited for about 12 weeks when humanization achieved, and then AML cells were injected intrafemoral bone. This way, we set human donor cells and the patient AML cells into the NSG mice, building the GVR model. This shows the first step, establish humanized mice. After HSC injection, serial blood samples were collected to measure the engraftment of human CD45 positive cells. This is a marker for hematopoietic cells. Panel A shows the representing flow cytometry data for human CD45 gating. Panel B shows the percentage of human cells over time in the blood. Significant number of human CD45 positive cells were detected in the blood at 4 weeks, and the number declined thereafter, but stabilized at week 12 and was sustained about 5% after at least week 24. At 24 weeks, mice were euthanized and cells from liver, bone marrow, and spleen were analyzed. Shown in panel C here, human CD45 positive cells were more frequent in these organs, with highest in bone marrow. To confirm the reproducible, we built a second model of humanized mice using another donor HSC, donor 2 here. We observed similar results. So we successfully generated humanized NSG mice by engrafting the donor GCSF mobilized HSC into the NSG mice. We next evaluate the development of human immune system in these humanized mice. Innate immune cells developed early in the mice with relatively high frequency of NK and monocytes in the blood at 4 weeks. The number decreased thereafter. In contrast, for T cells, there's a minimal number at 4 weeks, but significant increase at 12 weeks and remain above 30% at week 24. We also observed significant B cell development in this model. Adequate development of all the human immune component were also observed in bone marrow, spleen, and liver. This is the data at week 24 post-injection. We then focus on human T cell development in these mice. The CD4 versus CD8 ratio was initially reversed at week 12 and week 16, less than 1. This is consistent with what we observed in clinical transplant. CD4 development is delayed than CD8 after the transplant. So this ratio was increased to normal range above 2 at 24 weeks post-injection. When differentiation status was evaluated based on the expression of CD45RA versus CCR7 in T cells, we found a differentiation stage including naive central memory, effector memory, and terminal differentiate T cells in both CD4 and CD8 T cells, with terminal differentiate T cells are very minimal in CD4 cells. So these cells are very functional, shown by proliferation marker Ki67 and killing capacity, a bunch of expression of granzyme B, as well as cytokine production. Shown here in I panel, the intracellular production of interferon gamma and TNF-alpha by spleen cells upon in vitro stimulation with CD3 and CD28 antibodies. In addition, these T cells expressed a significant level of activation markers on the T cells. Shows here are human CD28, CD226, and CD30 expression. So there's an adequate reconstitution of human immune system, including functional T cells in the humanized mice. So I'd like to point out here that donor 1 HSCs were collected from a transplant donor, and that the cells were actually applied to an AML patient during transplant. We had followed this patient clinically and collected serial blood samples post-transplant. So this allowed us to directly compare the same donor HSCs in patient and in NSG mice. Consistent with our findings in the NSG mice, humanized mice, we found a reconstitution of innate immune cells early post-transplant. So both NK and monocytes developed significantly at 1 month post-transplant and trended down later on. In contrast, T cells frequency trended up over time. We further observed a low percentage of CD4 T cells and a reversed CD4 versus CD8 ratio until at least 2 years post-transplant in this patient. This is consistent with the early phase in the immune reconstitution in humanized mice. In addition, we observed a similar pattern of T cell differentiation status in that all differentiation stages were presented with low frequency of terminal differentiated cells in CD4 T cells. We also found that both CD4 and CD8 T cells was highly functional, showed by adequate Ki67 expression, showing proliferation, and the expression of granzyme B and porphyrin, which are representing the killing capacity, as well as production of interferon gamma and TNF-alpha upon in vitro stimulation. And also we detected expression of majority of activation markers on the T cells, shows in panel E here. So taken together, this data show that the immune reconstitution of human HSCs in NSG mice largely recapitulates the clinical scenario in the patient received the same HSCs during allotransplant. Importantly, we observed a similar pattern of T cell development, differentiation, and functional status in the humanized mice compared with that in the patient. This is a summary of this portion of the data. I'd like to point out here that we selected G-CSF mobilized peripheral blood from a transplant donor as a source for HSC for building this humanized mice. There's some privilege using this G-CSF mobilized peripheral blood. So compared with cord blood, this G-CSF mobilized peripheral blood are more widely used in clinical allotransplant. So this make our data more reflecting of the clinical scenario and mirror the clinical situations in patients. Second, the cell number is limited in each cord blood unit, but abundant in G-CSF mobilized peripheral blood, allow a study of patient-specific conditions. So with these extra cells available, we are able to find the immune reconstitutions of G-CSF mobilized HSCs in NSG mice, recapitulate some of the clinical scenario in the patient receiving the same HSCs during the allotransplant. So we showed similar pattern of the T cell development, differentiation, and functional status. We also showed significant reconstitution of NK monocytes and granulocytes in these innate immune systems. Compared with other mouse strain, this might be a little suboptimal. So if we want to look at focus on these cells, NSG, SGM3, and Mr. G mass might be more optimal. So far, we were able to show that using G-CSF mobilized donor PBMCs, we were able to build up humanized mice. So next step is to inject patient-derived AML cells into these humanized mice to build the GVR model. So just some characteristics of the donor HSC and AML pairs in the cells we used. So we use HLA partial matched pairs to prevent the rejection. So listed here are HLA match status for these two pairs. 6 out of 8 in pair 1 and 5 out of 8 matched in pair 2. So again, green box here is HLA match allele. The white box is HLA match. So on the right part is the clinical characteristics of this patient who provide the AML cells. So both patients are in their age of 50s. At the newly diagnosis, the WBC was very high, with majority of the cells are leukemia blast in blood. And cytogenetic features, there's some difference between the two patients. And the leukemia immunophenotype, we showed both patients leukemia expressed CD33 positive. This is very important because later on we are going to use this as a marker to pick out the AML cells. So patient-derived AML cells were injected into these humanized mice. Panel A shows the representative flow cytometry data for gating of the leukemia by CD33 expression. So in both models, leukemia blasts were detected in blood at 4 weeks, which increased up to 70% at week 12. This is in blood again. In contrast, the humanized mice alone without AML injection, there's a minimal CD33 positive cells. These are mostly progenitor cells. Mice was euthanized at 12 weeks post-AML injection to evaluate leukemia infiltrating into the organs. Consistent with clinical observation that bone marrow is the major anatopic location for leukemia, most human cells in bone marrow were CD33 positive leukemia blast. We also detect a significant number of blasts in spleen, liver, and lungs. Red skin cell staining confirmed the morphological features of blasts similar to that of clinical samples from AML patients, shown in panel D here. Also, we consistently observed enlarged spleen in humanized NSG mice together with AML, compared with humanized mice alone. So this shows represent the failure, secondary to failure of AML, and also leukemia infiltrate into the spleen. So taken together, our data demonstrate a growth of patient-derived AML into the humanized mice. To determine whether the reconstituted human immune system in the humanized mice has an anti-leukemia effect, we did another experiment injecting the patient-derived AML cells into humanized mice versus NSG mice without humanization. So human CD45, CD33 positive, double positive cells were gated to assess the leukemia burden. So panel A shows the data from the blood. At all timing point tested, the leukemia growth tended to be less in humanized mice compared with NSG mice alone. This was statistically significant at 12 weeks. So this was observed in both models. Consistently, leukemia infiltrating organs, especially bone marrow, was significantly lower in humanized NSG mice, shown in panel B here. We also evaluated CBC in the mice. Humanized NSG mice without AML, shows as a control here, has normal WBC, hemoglobin, and and platelets as well. And for NSG mice with AML, they have a much elevated WBC, majority of which are the leukemia blast, and their hemoglobin and platelet was dropped due to marrow suppression secondary to high leukemia burden. The number of humanized mice with AML were in the middle, as they do have leukemia growth, but the tumor burden is less than that in NSG alone with the AML. So a summary of this part, HLA partially matched patient-derived AML can grow in humanized NSG mice. We found significantly reduced leukemia growth and subsequently less level of bone marrow failure in humanized mice compared with that in control NSG mice. So this demonstrates a functional GVL effect. We therefore term these mice as a humanized GVR mice. Humanized GVR model provides a very good platform to study human GVR. So we have a humanized GVR mice here containing both donor-derived HSC, donor-derived human immune system, as well as patient-derived AML cells. If we compare these mice with that in humanized mice without AML, we can evaluate the influence of human AML into the human immune response. On the other hand, if we compare humanized GVR model with the NSG mice alone without immunization injected with AML, we can evaluate the effect of human immune pressure on the human AML cells. So now a little bit background here. It has been become clear that AML relapse post-allotransplant is largely due to the escape of leukemia from alloreactive immune response. So the mechanism that mediates leukemia immune escape during the GVH reaction, in other words, causing GVR failure, is not totally clear at this point. Several hypotheses were suggested by recent studies. First, loss of HLA class II expression on leukemia cells. Second, upregulation of immune checkpoint pathways. Third, production of inhibitory enzymes such as IDO, and also modulation of anti- or pro-inflammatory cytokines. And finally, increase of suppressive immune cells such as Treg or MDSCs. Among all these, the first two are more developed in this field. In terms of upregulation of immune checkpoint in relapse post-allotransplant, multiple studies, including our own, have shown that PD-1 and TIGIT, which is another immune checkpoint functioning in tumor, are upregulated and associated with the leukemia relapse in patients post-allogeneic stem cell transplant. To evaluate whether our novel humanized GVR model recapitulates these clinical features, we first assessed the expression of PD-1 and TIGIT expression on the CD8 T cells in humanized GVR model versus humanized NSG mice. We found that the frequencies of PD-1 and PD-1 TIGIT double positive T cells were significantly higher in humanized GVR mice compared to that in humanized NSG mice. We next test the expression of PD-L1, which is a PD-1 ligand, and CD155, which is a TIGIT ligand, on AML cells in humanized GVR model versus NSG mice alone with AML injection. So this NSG mice is without humanization. We found a significant increase of PD-L1 and CD155 expression on AML cells in humanized GVR mice. So consistent with clinical AML relapse post-allotransplant, humanized GVR mice has increased of inhibitory pathways under the immune pressure. Loss of HLA expression is another important mechanism for AML relapse post-transplant. This is supported by several elegant studies using clinical samples from allotransplant. These papers were published in last several years in New England Journal of Medicine and Nature Medicine. So we therefore test the HLA expression in our humanized GVR model and found a significant decrease of HLA-DR on AML from humanized GVR model compared with that on AML from NSG mice. So this is consistent with the clinical observation that loss of HLA correlated with AML relapse post-allotransplant. So a summary of this part, humanized GVR model can recapitulate clinical features for leukemia immune evasion in AML relapse post-allogeneic stem cell transplant. So in conclusion, we successfully established a humanized GVR model using human HSCs and HLA partially matched human AML. And we demonstrated a functional but defect GVR effect in this model. Humanized GVR mice recapitulate clinical features of leukemia immune evasion in clinical AML relapse post-transplant. We feel like this novel clinical relevant model system forms a very good base for studies of mechanisms in human AML, in human GVL, and test the novel agents in this model. So this is my acknowledged slide. Very importantly, we thank all our leukemia patients and their family for strong support and willingness to donate the clinical samples for our research. This work is mainly conducted by Bei, a postdoc in the lab who is extremely bright, dedicated, and capable scientist. Chen is another postdoc in the lab, also helped a lot in this project. My clinical colleagues, Dr. Claxton, Dr. Amon, Rebecca, Dr. Ho, Manishi, Naik, Dr. Work, Dr. Rizalski, they all helped very much in this project by consenting patient and make it available, possible for us to collect the patient samples. I'm also grateful to our collaborations, both outside and inside Penn State Hershey. Special thanks to Dr. Schutz, who has pioneered the generation of humanized mice and their use in multiple fields, including cancer research. We are very fortunate to have collaborated with him on these projects, as well as other ongoing studies. And finally, listed here is the funding support for this study. We are very grateful for all the strong support around, allow us to make advance in our leukemia research. Thank you very much for your attention.